I recognize that our audience is quite diverse in terms of backgrounds, and I know we have people with general scientific backgrounds, neuroscientists, the people in the public who are interested. So I'm trying to give you something for everybody, and I probably will fail miserably, but let's give it a try. So I wanted to start with some background and talk a little bit about heart disease. There's a connection, of course, between the heart and the brain, but I really want to point out the tremendous success we've had in this country in dealing with heart disease on a public health level. And what I first want to point out is, if you look at this graph, you'll see that there's a period of increase here when heart disease was just getting more and more prevalent, and we attribute that to people living longer, to diets that weren't really the best, especially to smoking, to a sedentary lifestyle, and recognition of things like heart attacks. And then things started to change when people began to collect data in the late 1940s. The Framingham Heart Study that you probably heard about began to collect a lot of data and began to collect information about risk factors like smoking, cholesterol, and hypertension. The clinical trials were started to lower hypertension, to lower cholesterol. Drugs became available like thiazides to lower blood pressure and lovastatin to lower cholesterol. And we began to see this tremendous decrease over the last 30 or 40 years, probably related to decreases in smoking, to treatment of hypertension and hyperlipidemias and so forth. And this has really been a tremendous success story. Even though cardiovascular disease is still the #1 killer in our country, it's really improved. And I just want to point out that this has been going on for 70 years, and it's taken 70 years, and we have to do better than that with Alzheimer's disease. This is the funding from the National Institute on Aging over time, and what you can see is the funding for cancer in purple versus the funding for Alzheimer's disease. And only recently has funding for Alzheimer's disease really started to increase. So this is something, excuse me, this is something that helps drive innovation. It helps drive clinical trials. And I think many of us in the field are beginning to feel optimistic. But I just would point out that it's going to take a lot of time and a lot of resources to really solve this problem. Okay, so I want to start out by talking about the neuroscience of memory, and I'm going to do this very quickly just by mentioning probably one of the most famous patients in the history of medicine. His initials were HM. He died in 2008 and actually had an obituary on the front page of the New York Times. And he was famous because he underwent an operation to treat his epilepsy, in which part of the temporal lobe was removed. And it's right here in the medial part of the temporal lobe. It was removed to treat his epilepsy. And after the operation, he lost all ability to form new memories. What happened is that, for example, he had a number of psychologists who worked with him daily. Each time the psychologist would come into his room, she had to introduce herself again. He couldn't remember who she was. So he had the inability to form new memories, and it was obviously a sad story, but it was a very important story to our understanding of memory because it implicated these structures in the brain, in the medial temporal lobe, including particularly the hippocampus. It implicated these structures in the formation of memory. And part of the story that I'm going to tell you today is about how these structures are affected by aging and by Alzheimer's disease. So just again, to put everyone on the same page, Alzheimer's disease is the most common cause of dementia. This is sort of meant to give you an idea. Alzheimer's is one cause of dementia. It's not the only cause of dementia. Dementia is a loss of ability to take care of oneself. It's a loss of cognitive ability. And Alzheimer's disease is far and away the most common cause. It involves a progressive loss of cognitive ability that interferes with daily life, as I said. Now, Alzheimer's disease is particularly unusual in two ways. There's a very typical neuropathology that I'm going to talk about. It forms the basis of our understanding of Alzheimer's, this neuropathology, and it affects vulnerable brain regions and affects vulnerable systems in the brain. And particularly, it begins with a loss of the same kind of memory that I described in HM. That's the problem with what we call episodic memory, which is an inability to remember the events of one's daily life. So for example, you may forget what you had for breakfast. You may forget a person you met. You may forget, in the clinic, we test this by giving people lists of words to remember. This isn't long-term remote memory from childhood. This is memory of what happened to you a few minutes ago or a few hours ago. Now, I've seen some smiles in the audience. Because who doesn't have that, especially once you get to be over, 40, maybe even younger. So that's another part of the story that we're going to talk about today. Some loss of memory is pretty common. This type of memory loss is not rare at all. But when people with Alzheimer's disease experience it, it's quite severe, and it interferes with an inability to function in daily life. And so this is the fundamental memory problem of people who have Alzheimer's disease. So we understand the disease by looking at the brain pathology. And what I'm showing you here are two pathological abnormalities in the brains of people with Alzheimer's disease. One of them is the plaque, and it's composed of this material called beta amyloid. It's interstitial, meaning that it's between the cells in the brain. And this is a beta amyloid plaque. And then there's the tau, another protein. So one protein is beta amyloid, the other protein is tau, and tau is in a structure that's called the neurofibrillary tangle. So plaques and tangles are the pathology of Alzheimer's disease, and when we see these pathologies in the brain in sufficient amounts, pretty high amounts actually, in an individual who suffered from dementia during life, we generally conclude that that person had Alzheimer's disease. Now, it's not that simple. It turns out that tau is deposited in the brain in a very specific region, that red area there that's popped out, and that is the medial temporal lobe, and that includes the hippocampus and other parts of the temporal lobe that are connected to the hippocampus and that are involved in the memory system. So tau deposits here in the context of aging. As we get older, we start to see tau in this particular brain region. The amyloid plaques are different. They're deposited throughout the brain. So the location of these two proteins in the brain is very specific, very interesting, very different, and it's telling us something about the disease of Alzheimer's disease that I have to confess we don't fully understand. But what I want to show you is how common these pathologies are with age. This is simply age by the percentage of people showing this pathology, and especially these purple lines, which are the tau neurofibrillary tangles. They're virtually ubiquitous in the medial temporal lobe memory system by the time we get into our late 60s and 70s. I'm the same age, by the way, so I probably have some. So we can see these. We can see these in the brain, we can see the tau pathology, and we can see the amyloid pathology with PET scans. And in order to do a PET scan, you have to start with a cyclotron. So that makes it kind of complicated. And the cyclotron is necessary to produce the radioactivity that enables us to see the compounds. So we take this cyclotron, we create a radioactive atom, and we hook it on to a molecule that we're interested in a radiochemical synthesis, and that molecule is super interesting because we design these molecules to bind to the proteins in the brain. So they bind to the beta amyloid or they bind to the tau, and the radioactivity from the cyclotron allows us to see where it goes in the patient, in the body and in the brain. So we simply inject it into the individual, we see where it goes, we put the individual into a PET scanner, which makes a map of the distribution of this radioactivity. And because we understand how the compound binds, we can make a picture of where the radioactivity is, and that will tell us, for example, where the amyloid is, where the tau is, and potentially a whole variety of other kinds of biochemical processes in the brain. So there's really a tremendous number of different processes we can visualize with this approach. So in the story that I'm going to tell you has really been moved along incredibly quickly by the innovations that are related to this technology. And the first thing that actually we had available showed up about maybe 20 years ago. I guess that's old. That's a long time ago, but I remember it very, very vividly because the first PET tracer for amyloid was a real breakthrough. So we didn't do this in my lab. This was done by investigators actually at the University of Pittsburgh who invented a compound that's radioactive, injected into the person, and binds to the amyloid in the brain. And what you're seeing here is a map of where the amyloid is in this particular individual. Now, it turns out we can also measure amyloid in the spinal fluid with a spinal tap, but that doesn't tell us where the amyloid is in the brain. About 6 or 7 years ago, new tracers came along for tau. So now we can image the tau as well. Same idea. This is a radioactive compound injected, and we're seeing a map of where the tau is in the brain. And then we have other ways of looking at what happens in Alzheimer's disease and aging that we call neurodegeneration. And that's basically shrinkage of the brain and dysfunction of the brain. So we can see that with MRI scans. So with MRI scans, for example, that arrow is pointing to the hippocampus. And we can see the medial temporal lobe, those structures that are involved in episodic memory. We can see those structures as they shrink, and we can measure that as an outcome of these amyloid and tau pathological processes. We can also look at glucose metabolism in the brain, which has a very specific pattern as well in association with Alzheimer's disease. So I'm going to show you some scans of all of these tracers, all of these biochemical processes in a single person, just to point out sort of how they're the same and how they're different. So as I told you at the very beginning, the amyloid, so in these, all the images that I've shown you, I guess I didn't explain this, the bright, hot colors like reds, especially yellows, lesser, are where the amyloid or tau is. So you can see the amyloid in this scan is pretty much everywhere throughout the brain with some important exceptions. But the tau is really much more located in the temporal lobe and the parietal lobe, and that parallels the changes in metabolism and brain shrinkage or atrophy that are part of this neurodegeneration process. So the neurodegeneration process does not follow the amyloid as much as it follows the tau, and we're going to come back to that. So I'm going to take a little digression and talk about the molecule of amyloid, because I want you to understand why we're so obsessed with this protein. So the amyloid protein is a very large, protein that it spans the membrane of the cell. But the important part of the story is this large protein, which we call an amyloid precursor protein, is cleaved by two different enzymes, a beta secretase and a gamma secretase. And when that happens, this part of this large protein is freed up and travels out of the membrane, and that's the A-beta, or the beta amyloid that I've been talking about. So it's a cleavage product from this larger protein, and it aggregates and it forms these plaques. Now, we don't have proof for the next part, but we think that somehow there's an interaction that results in the tau as a secondary phenomenon, and I'll show you why we think that. So what's all the fuss? So let's go back to this particular protein, the beta amyloid, the gamma secretase, the beta secretase. It turns out there are families that have mutations in this whole metabolic pathway, starting from the protein itself to the enzymes that cleave it. And these mutations are very rare, they're quite uncommon, but they're really important because of what they've told us about the role of this particular protein. And so, for example, individuals with mutations in this amyloid precursor protein have mutations right around the sites where the beta and gamma secretase cleave the molecule. And other mutations occur in parts of this gamma secretase enzyme as well. These are the presenilin mutations. And why these are important is because all of these mutations are associated with increased production of this plaque-forming beta amyloid. So if you have a mutation in one of these genes, you will get Alzheimer's disease. Thankfully, it's extremely rare. You will get Alzheimer's disease, and as far as we understand it, it's because the individuals with these mutations overproduce beta amyloid because that's what the mutations do. So this has been a very, very strong link between amyloid and Alzheimer's disease. It's sufficient to have these mutations to get the whole picture of amyloid. You get the amyloid and the tau with just these amyloid mutations. So I'm going to tell you about a really interesting study that I'm not part of. It's called the Dominantly Inherited Alzheimer's Network, or DIAN. It's being conducted by investigators at Washington University in St. Louis. And what they've done is they've recruited a few hundred people with these gene mutations. And as I said, these are very rare mutations, so they've created a network that's international, and they bring people into the laboratory to study them. And what they've done is they've measured all the biomarkers that I've been telling you about. They measure amyloid, they measure tau, they measure brain degeneration. And the interesting thing about this particular study is they were able to recruit people who have the genetic mutations and compare them to family members who don't have the genetic mutations. And what's also really interesting about this particular study is that the age at which people get sick is relatively preserved from generation to generation. So, for example, if you are unfortunate to have one of these mutations, your mother had Alzheimer's disease when she was 50, the chances are that you're going to get Alzheimer's when you're 50, plus or minus a few years. So what they did very cleverly is they brought people into the laboratory, they did PET scans, they characterized how far away they were from their estimated onset of the disease. So if your mother if you had a parent who got sick when she was 50, you had a PET scan at 40, then you're 10 years before your likely onset of disease. And what they did is they mapped out the changes in biomarkers. And that's what you're seeing here. And in fact, it's maybe a little hard to see, but the first thing that changes is this beta amyloid in the brain. And this beta amyloid here is starting to go up in the brain of people who were 20 years before they were expected to get sick. And then what you start to see is other things changing, like the brain, the brain shrinking, glucose metabolism going down, et cetera. So this is a 20-year process, and at least in the autosomal dominant cases, this begins with amyloid. And this has led to the amyloid hypothesis, which is that amyloid deposition is the initiating event in Alzheimer's, leading to these tau tangles and brain degeneration and dementia. Okay, so now let's talk about some of the science that's grown out of this idea. So this is a PET scan of a patient with Alzheimer's disease, and I've explained to you that these hot red areas are where the amyloid is in the brain. In my lab, we're studying mostly normal, healthy, older people because we want to understand the transition in the very, very earliest stages of Alzheimer's disease. So we do study some Alzheimer's patients, but we're really focused on recruiting a cohort of individuals very similar to what's happening here in Dallas, of people who are older and at risk of developing Alzheimer's disease. So this is a patient with Alzheimer's, and this is one of the participants in our normal aging study. And you're going to have to take my word for this, but there's no amyloid in this person's brain. The green-blue colors are sort of a background level of binding. There's no amyloid in this person's brain. So that's what we expected. But look at this individual. This is also a normal, older individual. And actually, this normal, older individual has a lot of amyloid in his or her brain, just as much as the Alzheimer patient. I can tell you that you wouldn't be able to tell these two normal, older subjects apart, the one with amyloid and the one without amyloid. I'm confident that you couldn't tell them apart because we can't tell them apart by giving them cognitive tests. We really are not sure. So this amyloid is not having a big effect in these individuals because they're still cognitively normal. And this is a very common story. So maybe a little more complicated graph here. This shows age, and this is the amount of amyloid in the brain. These are a group of people in their 20s who are our true control subjects because we know they don't have any amyloid. They all have a value here of about 1, which means no amyloid. These are our older controls. They mostly are around 1. The light blue is our Alzheimer patients. And what you see is that many of the controls in purple have just as much amyloid in their brain as the Alzheimer patients. And this isn't a finding, it's finding from my lab, but this has been found around the world in most studies. 20 to 30% of cognitively normal people in their 70s have a substantial amount of amyloid in the brain. So what are we going to make of this? Here's the amyloid. Here's the relation to cognition. And it's really weak. It's not strong. Well, you know, I left all that black space in the slide for a reason, because other things are happening. And this is what is happening. And this is, in fact, the chain of events that we think is occurring, that amyloid is doing something to drive tau, tau is doing something to drive neurodegeneration, and that's doing something to drive the cognitive deterioration that I've been talking about. So let's talk about the amyloid and tau relationship. Just show you one study that we did. This is a relationship between the age of the participants in our study and where the tau is in the brain. And what you see is that as people get older, the tau increases in the medial temporal lobe. So this was the first PET study that we did with this tau PET scanning, and we were very happy to see this because it's what we thought we should see based on the pathology data that I started out by showing you. So older people have more tau in their medial temporal lobe. What happens when amyloid comes along is that the tau spreads out of the medial temporal lobe, and you start seeing it throughout the brain. And so amyloid is somehow driving the spread of tau out of this medial temporal lobe brain region. So how about this part of the story? So this is a study we did with our collaborators at UCSF, which everyone had a baseline scan, and then we looked at the rate of brain shrinkage. And they had a baseline scan for amyloid, for tau, and for MRI, cortical brain volume. And what you can see here is that, yeah, people with more amyloid in the brain showed more shrinkage of the brain over time. People who had smaller brains showed more shrinkage over time. But the real predictor of how much the brain shrank was the amount of tau in the brain. So the more tau individuals had, these are people with Alzheimer's now, the more tau they had in the brain, the faster their brain shrank over the next several years. In healthy older people, we actually see relationships as well. And this goes back to the comment I made early on about relationships between, about memory loss in healthy older people. And what we did in this study was something similar to the first slide I showed you, where we looked at the relationship here between memory and tau throughout the brain. And what we found is that as people's memory was worse, and in fact, as it declined over time, there was more tau in these particular brain regions, again, in these medial temporal lobe brain regions, which were really in this brain region called the entorhinal cortex, which is right next to the hippocampus. So the point I'm trying to make here is that the more tau in these brain regions in healthy older people, now these are not people necessarily who had amyloid in the brain. This is not people who were complaining to the doctor about symptoms. These are healthy older people. The more tau they had in these medial temporal lobe brain regions, the worse their memory was. Not terrible, not enough to result in a diagnosis, but enough that we could measure it. And then finally, I'll show you some scans in patients with Alzheimer's disease. These are amyloid PET scans. And in this study that was done by our colleagues at UCSF, there were three kinds of Alzheimer's patients. There were Alzheimer's patients who had memory loss, Alzheimer's patients who suffered from aphasia or language disorders, and Alzheimer's patients who had primary problems in visual ability. So these are unusual kinds of Alzheimer's. Most Alzheimer's begins with memory loss, but some Alzheimer's begins with speech difficulties, and some Alzheimer's begins with visuospatial problems. And what I want to show you here is that all three of these types of Alzheimer's disease have the exact same pattern of amyloid deposition. But that's not true for tau. The patients with typical Alzheimer's disease and memory loss actually have most of their tau in the parietal lobe and in the temporal lobe. The patients with aphasia, it may be a little hard to see, but this is the left hemisphere, this is the right hemisphere. There's more tau in the patients with language disorders in the left hemisphere, and we know that's responsible for most language function. And here, in visuospatial cases, there's much more tau in the posterior parts of the brain. Here, for example, compare these visuospatial cases to the other cases. There's more tau in these brain regions, and those are the parts of the brain that are involved in perception and and visual organization. So the pattern of tau correlates with Alzheimer's symptoms, but the pattern of amyloid doesn't. So I hope I've given you the idea that amyloid is sort of this non-specific, well, I shouldn't say non-specific, it's not specific in its deposition, but it somehow does something to tau to change it, and it's the tau that seems to be causing a lot of the trouble. So let me sort of put together the model, the next part of this model of what we think Alzheimer's disease is, and that is that older people accumulate tau in these medial temporal regions. That's what I'm trying to depict here. And it may be a cause of age-related memory loss that is probably pretty benign. At least it doesn't seem to be necessarily associated with, loss of independence or et cetera. And then when amyloid is deposited, this tau spreads out of the medial temporal lobe. And that's when problems really start to show up, because widespread tau deposition is associated with cognitive loss and dementia. In fact, you can have lots and lots of amyloid in the brain, as I've shown you, and be completely cognitively normal, but you can't have lots and lots of tau in the brain and be completely cognitively normal. It would be very unusual to see that. And so we think, in a way, this is the beginning of Alzheimer's disease, the spread of tau. Now, I haven't told you a lot, and mostly that's because I don't know a lot. We don't know why tau is deposited in the entorhinal cortex in normal aging to begin with, and we don't understand what amyloid does to tau to make this process happen. And those are two really big holes in our knowledge, because those are potential things that we could do. Those are potential targets for therapies, for example. But I just have to tell you, we don't understand those processes. I'm going to take a little side trip, though, to tell you something that I think we do understand better now than we did maybe 5 years ago, and that's why the tau ends up where it is and how it sort of gets there. And this is a little neuroscience-y maybe, but I think everyone can follow this story. So I've already kind of shown you this, that the amyloid has one distribution in the brain, and it's sort of, especially throughout the cortex, it's in these parietal and medial frontal regions particularly, and the tau is in the temporal lobe particularly, especially early on in the disease, and that's what we're trying to understand, is what's happening at the beginning of this process. And so to sort of tell this story, I have to get a little more involved in the story of memory, and I've been talking about the medial temporal lobe as being sort of the memory system in the brain, but there's actually sort of there's at least two components to this memory system, and one of them is what we call an anterior temporal system, or anterior temporal network, I should say, which is involved in memory for objects. And then there's a posteromedial network, which is involved in memory for scenes. And so we think these two parts of these networks work together, probably come together in the hippocampus, where they seem to be able to bind information to create memories. So why am I going into this? Well, I'm going into it because if you really get into the part of the brain that the tau deposits, which is the entorhinal cortex, there's a very specific area that we think the tau begins, and that's in this area where this arrow is. This is, for those of you who want to follow this is a coronal section, I think maybe you know, and it's showing you the entorhinal cortex here and the periorhinal cortex here, and this region at the boundary of the entorhinal cortex and periorhinal cortex has been called transentorhinal cortex, and that's where we think the tau begins, and it's actually in the anterolateral part of the entorhinal cortex, which is part of the object anterior temporal system. And the posteromedial entorhinal cortex is here, and it's part of this posteromedial system. So these two systems are hooked up differently. And we thought this was a very interesting sort of set of findings that we could use to probe the question of how does tau get from where it starts, which is the anterolateral entorhinal cortex, to the rest of the brain. And our hypothesis was that it travels through brain connections. Now, I don't want to give you the idea that we came up with this hypothesis because there's lots of evidence that tau travels through brain connections. There's lots of evidence from experimental animals, and there's lots of evidence from humans, and we're certainly not the only ones to do this. But we were particularly interested in trying to figure out if this is how tau gets out of the entorhinal cortex. So to do that, we used a technique called functional MRI connectivity. And I don't want to go into the details, but in this particular technique, we can look at spontaneous fluctuations in the signal from an MRI. So this shows you the spontaneous fluctuations in one brain region, spontaneous fluctuations in another brain region, and we can look at the correlation between these two fluctuations, and if they're highly correlated, we conclude that these brain regions are connected to one another. So what we did, I shouldn't say we, because you all probably know I don't do any of this work. These are smart trainees. This was a graduate student in my lab named Jenna Adams who did this. I'm very proud of her. She started out looking at the anterolateral and posteromedial entorhinal cortex. She looked there and she asked what brain regions are connected to that. And this is what she found. She found this network of brain regions that's connected that looks a lot like where the tau deposits. And when she looked at the posteromedial, she found a completely different network, which looks like where the amyloid deposits. Now, before you get your hopes up, I don't know anything about this. I don't know how this happens. But this is pretty interesting, and just to confirm, these are really different networks. I think by now you keep track of the red as the anterolateral anterior object tau network, and you can see these networks don't really overlap. And it turned out that the more connected the brain region was to the anterolateral entorhinal cortex in red, this is the amount of tau in the brain. The more connected they were, the more tau there was. And the stronger the connection, the more tau there was. So we think that there's good evidence that tau ends up in this particular pattern in the brain because that's the part of the brain that's connected to the entorhinal cortex, and the amyloid is driving that tau out into the cortex. And just to point out, these are the people with amyloid in the brain, and these are people without amyloid in the brain. So there's a big effect of this difference in having amyloid in the brain. Okay. I'm just going to end by talking about what probably you're all very interested in. I guess I won't take a survey to see who hasn't heard of Adukanemab, but I bet a lot of you have. Adukanemab was a drug that was recently approved by the FDA. Even before it was approved, it was controversial. This article appeared in the New York Times several weeks before, I guess it was a little more than a week, before the FDA announced its decision and said, this isn't the drug that we want. The drug was approved, I think it was June 7th. June 7th. And it says, FDA approves Alzheimer's drug despite fierce debate over whether it works. And this is the Times story. And then, as you may know, advisors who were on the committee that gave advice to the FDA felt that their advice hadn't been listened to, so 3 of them quit. So people were really angry about this. And to this day, it's still controversial. The current status is it's approved by the FDA, but the Center for Medicare Services hasn't decided yet how or even if they will pay for it. So how do we get here? What's going on? So this was a really unusual clinical trial, and I don't know, you may not be interested in this, but it's kind of an amazing story, at least to me. So the trial was halted in 2019 because an advisory board looked at the data and said it's never going to work. So it was halted for futility. This was before the study was concluded. They looked at the data and said it's not worth exposing people to the risks of this drug. Let's stop the trial. But after they stopped the trial, data kept coming in. They weren't giving the drug anymore, but they were still getting data. And so they looked at the data, and one of the 2 trials they decided was positive. So they went to the FDA with this. The advisory panel voted against approval. 10 people on the panel voted no. one person voted uncertain. So no one on the advisory panel was in favor of approving the drug. The FDA approved it in a very unusual way. They actually didn't look at the clinical data. They didn't look at whether there was improvement or not. They looked at whether the drug lowered amyloid in the brain. And it did. I'll show you in a minute. It really lowered amyloid. It was a very potent drug in that way. And this is a pathway that was developed actually during the HIV/AIDS era that allowed drug makers to develop drugs based on things like T-cell numbers, for example. And it hastened the approval of HIV drugs, and it worked great because people didn't have to do a study to see how many people died from HIV. They could look at the effect on the T-cells. So it's a great idea, but there was a lot of controversy. Well, you lowered amyloid, but how do we know that's going to work? And there have already been some studies that failed, even though they lowered amyloid. So this is another rather minor thing, but it was approved for Alzheimer's disease, even though only patients with very mild Alzheimer's were allowed into the study. They originally came out with a list price of $56,000 a year. That's since been, to my understanding, it's cut in half, but it's still very expensive. And there are some allegations of whether there are inappropriate interactions between the FDA and the sponsor. So what does this drug look like? So here's PET scans. By now, you're all PET scan experts. These are the placebo. These are the baseline, sorry. This is the placebo. So you see over 1 year, no change. But as you increase the dose, you can see the amyloid is being cleared out of the brain on the PET scan. And there were 2 trials. One was called Emerge. The other was called Engage. They were huge trials, 1,500 participants with mild Alzheimer's. They got 1 of 2 doses or a placebo. And the drug, as I said, really, really lowered amyloid. This is the Emerge study. This is the placebo. This is time. You can see amyloid going down in both of these groups. So it really lowered amyloid more in the higher dose than the lower dose, which was a great result, made sense. Everyone was happy. Engage, very much the same thing. 2 clinical trials, both big effect on lowering brain amyloid. Here's the primary outcome, which was a clinical measure of cognitive and functional impairment. On this scale, higher is worse. Everyone got worse over time. The gray line is the placebo group. This is the low and high dose, and you can see that there actually was a significant difference between the high dose and the placebo. So this study, strictly speaking, was positive, although you can actually argue about how big an effect this was. It's not huge. You can see that with your naked eye, I think. It doesn't look like the amyloid graphs, does it? So that's the Emerge trial, but the Engage trial didn't show an effect. Those 3 lines, for all intents and purposes, are statistically identical and parallel. So did this drug work or not? Well, we can discuss it if you want later. People are fighting about it. I think no one would argue about these results. Everyone agrees. The real question is, should it be approved? Should it be given to patients with Alzheimer's disease? And I didn't tell you, the drug has side effects. It causes brain swelling in about 30 to 40% of people who get it, mostly very mild, but sometimes not. All right, I'm just going to leave you with one more data slide and tell you what I think one of the problems is. It's a little complicated slide, but I'm going to show you this and one more. And this is sort of the trajectory of amyloid deposition in a big study that I'm involved in that is sort of the rate of change in amyloid deposition in the brain. So we looked at people over time to look at how much amyloid is being deposited. And just to sort of simplify this, very early on, the rate of amyloid deposition is pretty fast. So higher is faster. So it's pretty fast. And then it sort of reaches a peak rate of amyloid deposition, and then it starts to slow down. So amyloid isn't deposited at the same rate over time. It seems to be very fast at first, and then it slows down. And you can take these data and mathematically rearrange things so that now you're looking at the amount of amyloid in the brain as a function of time. And all of these little red dots are sort of landmarks. So this is when you become positive for amyloid. This is when it reaches the peak rate. And these are the red dots that indicate the amount of amyloid in the brain of a typical person with Alzheimer's disease or mild cognitive impairment. So you can see it's going up over time. Well, two things are interesting here. One is that amyloid is accumulating for a long time before it starts to slow down. That's the peak, so it's accumulating at least 10 years before it starts to slow down. But then it's continuing to accumulate before people are impaired, but it starts to slow down even before people show up with Alzheimer's disease. So in other words, the rate of amyloid deposition seems to be slowing down before people get sick. So if you're trying to lower amyloid in people who are sick, maybe it's too late. And I think that's only one argument for why it's too late, because there's also the argument that once the damage has been done, it's too late. So this is, so we need to think about people getting treated even before they have symptoms. So how can we do that? Can we do PET scans on everyone on your 60th birthday, like you get a colonoscopy? I don't think so. A PET scan costs like several $1,000s. I don't know what a colonoscopy costs actually, but it seems like a pretty expensive and very useless approach. But here's some really interesting data. We can now, I say we, again, my lab doesn't do this, but there have been a number of papers measuring tau in the blood, in plasma. And this is one such study that looked at plasma tau in relationship to brain tau. And there's a pretty good prediction. It's not perfect, but if your plasma tau is high, there's a good chance that your brain tau is high. And this is a study of amyloid that shows pretty much the same thing. If you're above this cutoff for your PET scan is positive for amyloid, and it turns out that, that these individuals can be also detected by these low levels of CSF amyloid. We think that amyloid goes down when the PET scan becomes positive because the amyloid is coming out of the spinal fluid and going into the brain. So we can measure amyloid and tau in the plasma. So this is where I think we all think that I can't speak for everyone. Many people think this is headed. So for clinical trials, we could pick individuals who have elevated plasma, amyloid, or tau, and who are asymptomatic, because that's inexpensive and relatively straightforward. The technology is not completely there yet, so don't leave the conference thinking you want to get a blood test, because you can't get one, but it's still a research phase, but I think it will move into the clinic at some point. And then once you have the PET scan, the blood test, you can do a PET scan to see how much pathology there is. And I think what you really want is critical levels of tau, because you want enough tau that you know a person is developing Alzheimer's disease, but not so much tau that your hope for reducing it is futile. So the tau PET scan may be very important for staging a person. And then we treat them with amyloid-lowering drugs, or maybe in later stages, tau-lowering drugs. So just to conclude and remind you of this model we're talking about, we think this process begins with medial temporal lobe tau and aging. There's an amyloid deposited. There's some interaction that we truly don't understand, which leads to the spread of tau, and that leads to neurodegeneration and to cognitive decline. And so amyloid in the brain is actually probably the first stage of Alzheimer's disease, because I'm not sure that entorhinal cortex tau is really Alzheimer's disease. There's a poorly understood interaction that makes the tau malignant and drives it out into the rest of the brain. This is strongly related to cognitive loss. This process clearly develops over decades. And early lowering of amyloid is a reasonable target for therapy, but it's probably going to have to be early. And tau lowering may also play a role, especially later. So I'm going to end there. I kind of alluded to the fact that I don't really do any of this work. I have one of the joys of being in Berkeley is all the bright young people who are willing to work with me. This was our lab in April. We were pretty much on Zoom all the time, and then starting in July, we actually started to get out into the real world, and we're back in the lab. And things aren't going gangbusters, but they're better than they were. So thank you.
We're going to have Denise first. Thanks so much, Bill. That was tremendous, fantastic. Before we move to an open Q&A, Dr. Denise Park, who is on my right here, is going to give us a brief commentary on the lecture that we've just heard. Dr. Park is the founder and the founding director of the CVL. She's now the director of research at the center. She is a pioneer in the cognitive neuroscience of aging and has made seminal experimental and theoretical contributions to the field. We're delighted that she's here today, and we look forward to what she has to say by way of comment on the lecture that we've just heard. So Denise, over to you.
Okay, thank you. Thank you very much. I think it was very interesting to hear Bill talk about amyloid, because I think a lot of it's become a common belief that the amyloid hypothesis is dead and that there's no productive work coming from amyloid. My sister tells me this every day, and other people have mentioned this in some of our meetings. And I think Bill really did a wonderful job of showing you how complex the science is and how hard the decisions are. What you can get statistical significance, but you may not really improve the disease enough, and how much is enough? How much money should we spend for a modest improvement? And I think he clearly delineated why we think amyloid still matters. In fact, I can't wait to get to my lab. I have tested about 300 people. Some of you may have been subjects in our study with amyloid, all of normal, healthy aging. And we're finding, for instance, that, and Bill found the same thing, a similar thing in his lab, that there's one structure that we looked at, the posterior cortex, where posterior cingulate, where we found that if that's just one area where amyloid accumulates, and we find that even in middle-aged subjects, we can see that a little bit of accumulation in the posterior cingulate cortex results in a very small, almost undetectable change in memory, but it's real and it's there. And we think probably we're reaching a phase where we might be able to predict who's at risk 20 years beforehand. And as you can see from the talk Bill gave, that's going to be very important. The other thing that I want to mention is that it's not all genetic. The concordance rate for identical twins is 0.45. You have a 45% chance of getting Alzheimer's if your identical twin has it. So there's a lot going on. There's clearly a contribution from the environment, and we don't understand what that contribution is. But I think we need to, because we can control what we do to some extent. And I've wondered a lot about, I find that I can't sit still long enough to, if I get in a grocery line or something like that, what am I doing? I'm using my iPhone to play some silly game or a word-finding task or competing against myself or something. I think we keep our minds occupied and working all the time in this culture with the advent of these devices. And I'm beginning to think that maybe that plays a role in supporting our cognition. And the rate of Alzheimer's disease is decreasing slightly. So there's a little bit of good news. But the fact is, we need to figure out what it is that unlocks what contexts and what behaviors support the brain and may play a role in preventing Alzheimer's disease. And then I think Bill pointed out the technology we have is absolutely amazing. It's stunning to see what we can detect and what we can find. We can look into the brain, and even 15, 20 years ago, people couldn't even, all they could do was study Alzheimer's via autopsy. And we've come such a long way in that, and that's very exciting. I think the clinical trial work that Bill talked about is critically important, and I think he really delineated the politics. They're not exactly politics of science, because we were talking before this event, and there really were no bad guys here. It's not like somebody was really pushing that they were going to get a profit or something like that. It was a real legitimate argument about what is right, what works, what doesn't work. And I don't think there's any really bad guys here or good guys. They're just people that hold very strong opinions. And even when we have the data, we can't always decide what is correct or what is the best action to take. I think it's very important to study the healthy mind and to look at transitions, as Bill suggested. And I also think we need to invest more in studying middle-aged adults. I think the clues to healthy minds and to Alzheimer's disease are probably there in the middle age if we can figure that out. And I think that you should be able, I think the Tau is a very interesting issue as well. We have recently completed a collaboration, or almost complete. UTSW is making the Tau in their cyclotron for us, not the Tau, but the ligand that allows us to look at the Tau in the brain. And we've recently finished 100 people in our study that to look at the relationship between Tau and amyloid. So I'm eager to get back to my lab and very excited about the work that Bill presented, and want to thank him so much for joining us. Thank you, Bill.
Thank you so much, Denise. So yes, there's life in the amyloid hypothesis, amyloid hypothesis yet. So we're open for Q&A. The way we're going to work this is that we have people with microphones, I hope. Could they wave if they have the microphones? And if you could put your hand up, I will point at you. Someone will give you a microphone and you can ask your question. I'm just going to hand the mic over to Bill now. Bill, Denise, you're mic'd, and Bill, here's your microphone. And the first question, I can see a hand up just there. Marianne, could you do the honors? Could you just put your hand up so Marianne, just, yeah, there. Thank you.
Okay. I hope I read the graph correctly. It was, you show it at the beginning of the talk, and it was the deposition of beta amyloid comparing or contrasting 20-year-old versus healthy controls. And you pointed out the fact that some healthy controls have high accumulation. But if I read it right, there was some Alzheimer's cases that had the accumulation of a 20-year-old.
Yeah.
So what's going on there?
Very perceptive and absolutely correct. I'm going to stand because I feel like I'm going to fall over.
Yeah, I'm really nervous.
Yeah, you're totally right. And so these are misdiagnosed cases for the most part. Either they're cases with very little amyloid. So the reason I say they're misdiagnosed is because we now have a series. We had a paper about 2 or 3 years ago of 200 autopsy cases with clinical diagnoses. And if the individuals had high levels of amyloid, they had Alzheimer's disease. If they had low levels of Alzheimer's, they generally didn't. The sensitivity and specificity for Alzheimer's pathology with PET is pretty high. So, I mean, I can't tell you that every single one of these cases is misdiagnosed. I'm sure there are cases that are false negatives, but a lot of them were not diagnosed correctly.
Okay. More questions?
I regularly give blood. When you begin working on blood tests for tau and amyloid, is this something that you might have to test blood for so you don't pass it on in a donation?
No, there's no evidence that these proteins can be transmitted from one person to another by blood. That's a pretty straightforward answer.
Yeah, it's a good question, though.
It is a good question. Yeah, I mean, it's a little more interesting than that because these proteins do something in the brain where they sort of aggregate together. And theoretically, you could sort of make the argument that maybe this is happening, but there's really no evidence for it. People have looked.
Okay, We've got two questions. I'm going to take one on this side and then come back to here. So this one first, please.
Yes, a question and follow-up, I guess, on the drugs related to the one you showed was the results on amyloid. Obviously very positive from the picture, but doesn't tell the whole story or doesn't prevent the disease, as it turns out. So looking at that and then showing later the amount of amyloid, and you said it was kind of a baseline, and once it could get above that baseline, it might be a candidate for maybe diagnosis. How much of that drug, I got the impression the drug was almost completely eliminating it within 1 year, but what kind of, was it ever tested, like what kind of a small dose could keep it at a very low level for an extended period of time rather than go from a high level to zero? Could you just prevent it? And if so, and then is there something similar in the pipeline that could treat tau the same way?
Yeah, those are great questions. So the second question is easy to answer because the answer is yes. So these, I didn't really spend the time about how these drugs work, but what they are is they're antibodies. So they're antibodies. So it's a bit of a, the drugs that have worked for Alzheimer's, or that have lowered amyloid, I should say, because work or not is a question. But the drugs that have lowered amyloid are antibodies to amyloid that are infused into the bloodstream. So people getting these treatments have to come into an infusion clinic and get an IV. And they have to do it about every 6 weeks, roughly, or so. And there are drugs that do the same for tau that are being tested. So that's definitely an approach that people are trying for tau. These aren't the only approaches, by the way. There are many other drugs that are drug targets that people are looking at, but these are the ones that are the most advanced. So to your second question, it's a great question, and people are also looking at this. So there are studies where I think what's going to be, there are studies that are going to, for example, lower amyloid and then stop giving the drug, for example. So in other words, maybe you don't have to take this drug forever. Maybe you take it for a year, get your amyloid levels low, and then come back maybe in a year or 2 or 3 and see if your amyloid's gone back up. It probably will go back up, but it may not go back up really fast. So that's another approach. And yeah, I mean, I think the other question I think you asked, which I really can't answer, is, how low does it have to be? So a lot of these drugs, I mean, over the course of this whole approach, which has been going on now for, I don't know, 15 years maybe, of lowering amyloid, the drugs have just gotten more and more effective at lowering amyloid. And some of these drugs literally clear it entirely from the brain, in some people, right? Not in everybody. But the most recent drugs, the most recent generation of drugs can literally make the PET scan go from positive to negative. So can you get an effect from less of a dose than that? I mean, it's a great question. I don't think anyone is really brave enough to do that study right now, because we don't really see an effect from the big doses of lowering, right? But once we know that really lowering amyloid has an effect, maybe people will go back and see, well, maybe we can get away with something a little less.
Okay. We have a question here, Dr. Rosenberg, and then we'll go back over. Hold on.
Wonderful lecture. Thank you very much. I just wondered what your view is of inflammation late in the disease. When tau and amyloid have peaked, and microglia, the immune cell in the brain, becomes hyperactive and increases in number and puts out cytokines like TREM2, which seem to be inflammatory and cause neuronal loss. And just wondered what your view of inflammation was late in the disease. Thank you.
Yeah, so Dr. Rosenberg raises a really important part of the story that I didn't mention at all, because we haven't studied it. And I'm just gonna say the reason we haven't studied it is because we don't have good PET techniques really for doing that, because there are ways to do it, but it's not really optimal. But to your question, I think there's really strong evidence that inflammation may play a role early and late in the disease, right? I think at least some of the existing PET studies, which aren't perfect, suggest there may be changes in the inflammatory pathways even early in the disease. So this is clearly a target for therapy. There are, the original therapies were broad hammers to try to knock down inflammation, and there may be approaches that are much more targeted. So I think inflammation is it's not much of an answer, but inflammation is very important. I think everyone would agree with that.
Okay, we're going to take a question over here, and then we have a question here.
Thank you for the great talk, Professor. I just have a question about one of your results in your slide. So it looks like the brain region have a high functional connectivity with the entorhinal cortex. We're gonna have a high tau deposit. So I'm just wondering, except this functional connectivity, is there any like structural connectivity change between this, the cortical region and the entorhinal cortex? In the, like using DTI or other imaging study to see the structural change?
Yeah, we haven't done that. So there have been studies of structural connectivity in the whole brain that have looked at this. And I think, this story, I was trying to say we're not the first or the only people to make these links between tau spread and connections. And other labs have shown, if you look at a whole brain level, for example, both functional and structural connectivity are related to how tau seems to be spreading. So we didn't measure structural connectivity in my lab, though.
Thank you.
Okay, question here.
Yes, thank you so much for that lecture. So I'm a physician, and I'm here at Baylor. And as you know, every few years, Medicare throws us a bone and gives us the IDEA study, and I get to go from 4 or 5 PET scans a year to 100 a year, which is, I think, what I did last year. But I always have trepidation about someone like low 80s, mid 80s, about if they have cognitive impairment and they have a positive PET, how much to attribute. Is that contributing to their cognition, or is that aging? So I would just love your thoughts on that.
Yeah, so I didn't really go into one of the super complicated parts of this study, which gets to also the issue of people. There's two sides of your question. Does high amyloid always mean that Alzheimer's disease is going, or does low amyloid always mean that it's not Alzheimer's? And neither are 100% true. And that one of the reasons for that is that there are other processes that occur. And the most common one, for example, is stroke. So people who have small strokes or large strokes or multiple strokes can also develop cognitive decline. And there's some evidence that this works together with the amyloid. So there's two answers to your question. One is, just because the amyloid is there doesn't mean that is the only thing that's causing cognitive decline. The second one is, it's kind of semantic. And let me put it this way. I think if you have a lot of amyloid in the brain, you're very much at risk for Alzheimer's disease. I wouldn't call it Alzheimer's disease unless I knew there was also a lot of tau in the brain. So I kind of giving you a wishy-washy answer to your question. I think the problem is that that just knowing the amyloid status isn't enough to fully understand what's causing a patient's symptoms. You also have to know about other pathologies in the brain, stroke, et cetera, and also tau.
Okay. Thank you. We're going to take another couple of quick questions, and then we will bring the formal proceedings to an end. Yes, please.
I was curious if when they were testing the drug, did they have any way of measuring how much of it crossed the blood-brain barrier?
You know, I don't. I mean, I think their main measure of effect was the PET scan. I don't think they measured it in the spinal fluid. But I'm not 100% sure, to be honest with you. I don't think they did. But I think their measure of efficacy was the PET scan.
Okay, and there was. Okay, I think there was a question. Yes, please.
So I understand this probably isn't the area that you're focused on, but how much do we know about behavior change in prevention? Because I guess ultimately that's where we want to get to, right? So what do we know about the connection between behavior, environment, and I think Denise may have touched on that, but.
Yeah, I mean, that's just a huge question. Let me put it this way. There's a tremendous amount of observational data from epidemiological studies that link all kinds of exposures and behaviors to higher risk for Alzheimer's disease. These range from everything, and some of them are well replicated, others maybe not so much, but these range from everything from diet, exercise, staying cognitively active, to even things like a person's personality type. There have actually been pretty well replicated studies to suggest that conscientiousness, I don't know, you're all going to think about yourselves now, I do. people with high levels of conscientiousness have a lower risk of Alzheimer's and have a lower risk of Alzheimer's, and people with high levels of neuroticism have a higher risk of Alzheimer's. So conscientiousness and neuroticism are opposite poles on some of these behavior measures. I say this only because I think this is a pretty well replicated set of data. I don't know.
We're actually looking at that now. We have some.
Yeah, there's quite a few papers that have replicated this finding. I don't know what drives it. I think maybe conscientiousness might have to do with better health behaviors, for example, right? But there are a lot of behavioral factors, environmental exposures, and so forth that definitely affect the risk of Alzheimer's disease.
I was just going to ask, Denise, do you want to weigh in a little bit?
I would say that it should be clear to everybody that there are no large effects where if you do this, you're going to prevent Alzheimer's.
Right.
Even the effect of exercise is very, very small. It's there and it's real and it's reliable, but it's not huge. So you're not going to prevent Alzheimer's by doing one single thing or becoming very conscientious. It's a whole host of very complex interactions and complex behaviors and exposures that are going to resolve the issue. So we don't know yet what people need to do. And the brain is not that plastic as you get older. I suspect what you do when you're a child and what you do in middle age is much more important than what you do in old age.
I totally agree with that. I completely agree.
We always agree.
This is a life course issue, and it probably starts in utero, in my opinion.
Okay, I am going to bring formal proceedings to a close. Bill, I'm sure you will be happy to meet with people outside and chat further. I'd like, before we finish, to ask you all to join me in our appreciation again of a wonderful talk by Bill and also Denise as well, please.
Thank you.